I'm just going to try to project my voice and not use the microphone, so hopefully you'll be able to hear me, even in the back. This is the first of the research introductions to hopefully introduce the idea of doing research as an undergraduate and trying to hopefully get most of you involved in research as early as possible, because the sooner that you start research, the more likely you are to have results. And so Malcolm and I are the first two to go in this entire series. It's a year-long series from people from chemistry, biology, physics, computer science, and mathematics. I think we'll also see some. So today just doesn't have to be the first day that you do one of these things. So I'm Jonathan Dattelbaum. I'm in the Department of Chemistry. Why would you like to do research? Not why would you like to do research. Why do you do research? Yeah, but why would one like to do research?
To find out results.
Find out results. Find out results of something that you might be interested in. That's another reason for why one might do research.
To discover how nature works.
To discover how nature works. It's a very good example. Any other reasons why you might want to do research? Yeah.
Resume.
Resume, absolutely. Right. You know, that is a perfectly reasonable answer. Yes.
It's wicked sexy.
Wicked sexy. That is also a reason for doing research. Another along those resume building lines is also to help you develop some sort of contact with the faculty here at the university. Doing research is a wonderful way to get to know not only the person that you're working with, but because of the collaborative nature of a lot of the projects here at the University of Richmond, it's a great way to get to know lots of different types of faculty, and which goes along with helping resumes and letters of recommendation and things like that. So there's a lot of different reasons for doing research. I'll talk a little bit about what I do here, which is luminescent biosensors and sponges. And I went first on purpose so that I could steal a little of Malcolm's thunder, because I do a lot of stuff in the laboratory and he gets to go outside into nature and actually do a lot of cool stuff out of doors. So there was a purpose for me getting here early today. So I'm going to talk about my work with biosensors and with something about sponge pigments. A lot of my background is in fluorescent proteins. I'm a protein biochemist. So I'm also a fluorescent spectroscopist. I deal with the interaction of light with matter and being able to apply those things to different biochemical and biological systems. So I do a lot of fluorescent spectroscopy, a lot of molecular biology, protein biochemistry, and bioinformatics. Data mining, looking at the genomes of organisms for new proteins that you can characterize. And all of those things help me deal with the biosensor. So what's a biosensor? Here's a type of biosensor. This is a picture of a protein. It's actually the same protein. The only difference is that this little yellow guy is a ligand called ribose. It's a sugar. And when ribose binds to this protein, there's a large conformational change. If anybody ever has seen the game Pac-Man, right? Pac-Man is open and then he eats its closed. So this is one way to think about protein conformational changes. And let's say we wanted to be able to detect the sugar ribose. This little orange dot here represents a single amino acid. We can mutate that amino acid so that we can attach a fluorophore to it. When this conformational change happens, the protein alters conformation when it binds the ribose, and the fluorescence tells you the fluorescence signal changes so that you can detect when the ligand binds. This is a very simple type of sensing platform using fluorescence. This is just a diagram to show you. Here's your Pac-Man. These are types of proteins called paraplasmic binding proteins. They come from bacteria. And this is a fluorophore. And if any of you have taken organic chemistry, iodine is a very good leaving group. So iodine will leave and it will react with this sulfur group on the protein that you've engineered. So we actually do protein engineering to alter the amino acid sequence. And then that fluorophore gets detached. And then we can measure the protein's conformation. This is a fluorescent spectrum. So here you have fluorescence versus wavelength. And all we really care about, though, is the peak. In the absence of ribose, you see one level of fluorescence intensity. And then as you increase the amount of ribose, which in this case is this analyte, the thing that you're trying to measure. You see a decrease in fluorescence, and that's a measurable signal. You have created a biosensor. You've created something that can detect another molecule in solution. It's very sensitive, it's very specific, because proteins bind to ligands very sensitively and very specifically. One of the applications of this is in glucose sensing. You can have probably seen something that looks like this. It's a glucose meter, being able to take blood glucose levels. There's also other reasons to sense glucose in the pharmaceutical technological applications. These big vats can contain bacteria or some other growing organism, and glucose is a type of sugar, is also a major carbon source. And you have to monitor this throughout the growth cycle of these organisms, or your profits aren't as high. So there's a couple of different reasons. There's health benefits and commercial benefits to trying to come up with a glucose sensor. And one of my students, Brian Durr, I haven't really been mentioning my students. All the work that we do, of course, is done by people like you. One of my students, there's a glucose binding protein. It looks very much like the ribose binding protein, but instead, in this area right here, there are amino acids which aren't specific for ribose anymore. They're specific for glucose. But the same thing happens. You have an open form and a closed form, and this big conformational change takes place when the ligand binds. And I showed you before one way to measure the fluorescence change. This is another way. Instead of one fluorophore, we have two fluorophores. And when they're apart from each other, they have one level of signal intensity. And when they're brought together, they have another level of signal intensity. So that's giving us a measure of when glucose is binding to this protein to build a glucose biosensor. And how does it work? It works great. No glucose, and you increase glucose. The arrow is indicating increased glucose concentration. You see a rise in the fluorescence when glucose binds. So you're able to make a biosensor, and then over here on this side, you take these peak values and you plot them versus glucose concentration. This is called a standard curve. Some of you who are in my 141 general chemistry lab might have just seen an iron standard curve. It's exactly the same thing. You have a concentration of something that you're measuring versus a signal. Okay? So, and it turns out that this midpoint, the midpoint of that curve is very important. If you draw a line down from the midpoint, it's somewhere there. It's about 6 micromolar. That tells you how well this biosensor can respond, in what range it can respond. It's about 10-fold up, 10-fold back. Does anybody know what the concentration of glucose is in your blood? Any idea? Is it higher or lower than this? In any units, if you give me a different unit, if you happen to measure your blood glucose. No? So blood glucose, this is micromolar. Blood glucose is about 5 millimolar. So 3 orders of magnitude higher. And if you look at the scale, here's micromolar, there's 1 millimolar. So 5 millimolar is all the way out here. So you're at the very flat part of this curve if it was extended out. That's a problem for if you want to design a sensor because you have no fluorescence change anymore. So what we have to be able to do is shift this curve over into a usable region. And the way to do that is this is a blow-up of the binding pocket of that glucose binding protein. And these are the amino acids labeled in here. And so what some of my students did was they started mutating these things. They started using molecular biology, protein engineering to alter them so that this protein didn't bind glucose quite as well. It bound it very good. So 6 micromolar is better than 5 millimolar. And so we want to ease up. We want to make that a little looser of a binding. And when you do that, made a whole bunch of different mutants. This student, Brian Durr, they're all BD mutants. So he got up to more like 27. Made 27 of these mutants in my lab. And these are their KDs, these are their glucose binding constants. And here's blood glucose. And so one of them, out of all of them, one. This is another important point about research, is that you just have to keep going. Sometimes it doesn't work. These are all good glucose binders, but they're too good. We need to be up here. So found one that was good at binding glucose, and we were able to change its ability to more in line with blood glucose levels, a more practical type of application. One other thing I want to say about the fluorophores and how you do this. So I work a lot with light, and I'm using this green laser pointer. Green is very bright, and for some of you, this light will be so bright that you won't even want to look at it. It's very reflective off of the screen. Whereas red is not quite as bright. There's a reason for that. Your eyes just don't see it as well. When you're building biosensors, this type of phenomenon comes into play too. If you want to build a biosensor, it's finger pricking, right? That for diabetics, all day long they have to finger prick and draw blood from themselves. It's not a very pleasant process. If any of you like getting shots, you're probably in the minority. If you would think that you would like drawing your own blood 10, 12 times a day, probably not. And so when you're dealing with designing a biosensor, you want to think about light. If you look at this green, does it come through my finger? A little bit, maybe. That's because this is a really bright laser pointer. This one, again, is really dim. So don't blind anyone. How about now? The red really comes through. It turns out that your bone and tissue of everybody in this room is transparent to light that's red to infrared. And so when you're doing things like building biosensors, you want to have this type of idea in your mind. You need to understand light. You need to understand the properties of light. And you need to understand the properties of your skin and bone. So when we're building biosensors, you have to understand what the end application is. Now here's the part where I'm stealing Malcolm's thunder. I have two projects in the lab. The one was the biosensors, and Malcolm has brought me on to a project. It's now collaborative, looking at sponges in the Chesapeake Bay. He may even show you some of these pictures. This sponge, if I can use green and orange together, right? This is a Clathria prolifera. It's a common sponge found in the Chesapeake Bay. And we're interested in why it's orange and why some of those chemicals might be important for the sponge to be orange. And are some of those chemicals produced by the sponge or by organisms that live inside of the sponge? This is the experiment that got us started on this result. This is called thin layer chromatography. Basically, it's just a plate with a material on it. And what you do is you take extract from a sponge. You take a sponge and you throw it in methanol, and alcohol. You extract all the chemicals outside of that orange sponge. Even though it's orange, when you run them out on this process, you see all sorts of different colors. Not just orange, but greens and yellows and all sorts of different pigments that the sponge has. And you can look at it under white light or UV light. Difference. I'm a fluorescent spectroscopist. I'm interested in the differences in light, and that's how we got involved in this project, which is now running on 3 years and NSF funded, and we're still going with new ideas all the time. So one of the things that I was initially involved with was helping to characterize the chemicals that the sponge produces in response to different stimuli. And the stimuli is temperature. So what would happen if you grow the sponge in a tank at ambient temperature, or if you increase that temperature by a couple of degrees? And we run HPLC. It's another type of chromatographic separation. You can separate out all those yellow, blue, red chemicals that I showed you before. This is another way to separate them out. And when you do that, in blue is ambient in red is ambient temperature. And you see one pattern of trace. That's nice, but you need something to compare it to. So we compare it to the +2 degree trace, which is in blue, and you can see major differences in the profile. Major differences in the profile with just a 2 degree temperature change for the sponge. Is it good or bad? We don't know. We just know that there's a change, and so we're interested in trying to figure out what some of these chemicals are and trying to isolate them and characterize them. And that's an ongoing project. It's probably no longer called a new project. It's a new project for me because it's new for something that I'm doing as a protein biochemist. And we're using all sorts of different chemical techniques to try to isolate, characterize these chemicals. So that's my talk. I'll just acknowledge some of the current UR students. These are some Facebook pictures that they give me. And we've been able to be funded. And some of them are still here, some of them are gone. But research is usually a great way for students to get to know faculty and to learn something about nature and the environment. So thank you very much. I'll take any questions that you have. Oh yeah.
How do we go about getting into the research lab?
Great question. How do we go about getting into a research lab? There's a couple of different ways. The websites are a great place to start. Go to biology department, go to chemistry department. If you're interested in math, go to that department. Look at the websites. Just click on. Every person has a faculty link. So then just go and click and see if there's a little blurb about their research and see if it's interesting to you. If it's interesting to you, send them an email and say, I'm interested in research, where you talk for 5 minutes with me or something like that. Guarantee you get an email back and somebody will be able to talk to you. Yeah.
So you did a bunch of experiments over the last few years, and you gave the example with the carotenoid function and the expression. But what did you really find out that just changing the temperature affected the expression of the carotenoid?
It's a great question, and it's a subject of a paper that is in review right now. And I think you might talk about this.
I won't. I actually,
okay.
I had anticipated that you were going to talk about that, so that was actually good.
Oh, okay. The idea is that, please correct me if I'm wrong. The idea is that these chemicals, carotenoids, are photoprotective for the sponge. So the hypothesis is that the sponge makes these chemicals to defend against some environmental stimulus, whether that's temperature or light or some other predator, like something that actually eats sponges. So carotenoids they have a structure that's highly conjugated. Can't really see it.
No, a little bit.
A little bit.
That's green.
Yeah. They're highly conjugated structures. Draw my ring right. And what happens is singlet oxygen radicals are formed all the time. One of the reasons why our DNA gets damaged is from oxygen radicals forming. Sponges are the same way. They have a genome that can be damaged by oxygen radicals. And carotenoids are one of the chemicals that may be produced that may affect a defense mechanism for sponges. So in terms of, if the question is, what conclusion can we draw? Well, we can make small conclusions about the research. It's a work in progress.
Right, it's not yet.
That's right. We haven't made a definitive link that oxygen radicals are soaked up by carotenoids. But we can say that when temperature changes, we see a change in the carotenoid production. We can say when sponges are exposed to more light, which produces more oxygen radicals, we see a direct increase in the carotenoid concentration. So there's links there, but causation versus correlation is a tough
Thank you.
question. Any other questions? Yeah.
Technically, how did you figure out the conformational change of your chlorophyll?
Yeah. So the question was, how do you figure out that those proteins are undergoing conformational changes? The answer is structural work, X-ray crystallography, or NMR spectroscopy. NMR. And so Eugene Wu in the back here is a X-ray crystallographer type of person, and so you have to crystallize the protein, and that will allow you to identify the location of all the atoms in the protein, in the open state and in the closed state. And so that's actual direct experimental work. It takes a snapshot of the protein in each of those conformations. Anyone else? Okay. Then no other questions? Then we need to get the max set up.
I actually didn't mean, I was trying to be really inconspicuous and I wasn't like turning that off. Hey Michael, can you come down here? Seriously? I'm serious. See, I have this I have this rule in my lab that Michael's so new, he didn't get the memo, that if you come to this, I actually make you present for me.
I see you, sir.
Yeah, I've seen it. The last time I did this, I literally, 3 of my students came to this, and I said, okay, the best people to tell you about my research are these 3. And they literally the next day tried to kill me because they had no idea. Did you have any idea? I lied to you. Okay. Dr. Dattelbaum and I are collaborating and on a really interesting project that deals with the carotenoids that you mentioned. And we didn't, I purposely didn't add anything into this talk about that because I thought he might cover it. And he did. And it is really nice. It's a really nice and evolving piece of work that we don't have all the answers to, which is one of the really important things about science. When Dr. Dattelbaum was talking about why you want to do science, what are the things that make science science, I would add that it's organized play for adults. It's intellectual fun. And if you're not having fun doing science, then you're really doing something wrong. Because there are very few careers out there or opportunities, I think, to explore the natural world and figure out figure out how it works. And if you don't get jazzed up by that, then you're probably in the wrong place. But my sense is that most of you probably do. Or you wouldn't come at 5:30 at night to this room to listen to another lecture. You probably have that proclivity, which is good. Because science is organized play for adults. It's a lot of fun. If you're not enjoying it, then it's your fault. You're doing something wrong. Granted, there are tedious parts to the work, but the big questions, the fun stuff that you get to do in the lab or in the field, as I'll talk about in a minute, are just really neat, very neat. The other thing that, oh gosh, my computers may not survive this. I'm not sure why it's doing that. Oh wait, oh that's good. Do you want? The other thing that science is, that's really important is that it is a process. It's a way of thinking that's different than the way most humans think. It provides you with a set of skills, mental skills, intellectual skills, to ask questions, ask them in appropriate ways, and then answer them in satisfactory ways. So you can say, after having done all this work, I actually understand the system better. And that's a really important part of the process of going from entering the University of Richmond as a fresh high school student and going through the University of Richmond and going off to wherever you're going to end up going. During that process as science students, you get to think better about the way things work. And the thinking better part is the process of science, the methods that we use, the way that we think about and approach questions. So what I'm going to do, I'm going to go very quickly through some ongoing research that's the other side of the project that Dr. Dattelbaum didn't talk about. And then I'll let Michael, who I didn't know was going to be here. When I came in, I didn't see any of my students, and I was like, dang it. But then I'll let Michael talk about another project. And I almost wanted to pull Mallory down as well. Another project that's going on in the lab that's very, very different conceptually from the stuff that Jonathan and I are doing. Okay, so Jonathan didn't want to go first because I get to show pictures like, maybe. Maybe not. How does this work? Oh, this screen really hurt my eyes.
That's, yeah, right.
Okay, so my name's Malcolm Hill. I'm in the biology department. I'm a marine evolutionary ecologist, and I'm different than many of the other, for a lot of reasons, but I'm different than a lot of the other biologists that are out there because I do work in the field. Now, having said that, Dr. Carrie Wu, who's in the back, is a brand new faculty member who is also a field biologist, who does work out in the field. And we have some others in the department, Dr. Raphael Dessau, Dr. Peter Smallwood, among others, that do some work outside of the lab. And there are special and unique challenges associated with doing that kind of work. I also do work in the lab. It's not an either-or sort of situation, but the work that I do is, part of the work that I do is very different. And so I thought I'd focus on that because I thought Dr. Dattelbaum would talk about some of our work in Chesapeake Bay. I'm going to focus on some of the research that we do that's tropical, that's on coral reefs. We do a lot of our work at the Moat Marine Lab, which is down in the Florida Keys, on Summerlin Key, and we get wet. We scuba dive, we snorkel, and we work with sponges. That's our organism of choice. And we work with a particular type of sponge, one that harbors photosynthetic organisms. So it actually forms a symbiosis with plants. There's a recurring theme, one that unites the project that Dr. Dattelbaum was talking about and one that I'm going to present right now, and it's the theme of symbiosis. How do you get different entities living together? And how do they resolve conflict? Because those partners don't always have the same goals in mind. So for instance, you all have mitochondria, I hope, in your cells. Wouldn't be sitting here if you didn't. Sometimes your mitochondria don't have the same goal that you do in terms of living. Sometimes your mitochondria try to kill you. That's a breakdown in negotiation, cellular negotiation. It's a breakdown in conversation between these two entities. I'm really interested in that conflict piece. How do different species, different organelles, different things, how do they resolve conflict, evolutionarily and ecologically? So what I'm going to show you just very quickly are some images of work that we're doing trying to explain this question of symbiosis, and I'll throw a plug out right now. I'm going to be giving a seminar in the biology seminar series in February or March or January. I'm not sure when. But if you're interested in this at all, I encourage you to go. And that's not because I want you to come hear me talk. I actually want you to go to as many seminars as you can on campus. And this is I'm going to show you images now that I'm going to talk about again then. So I'm going to kind of just give a very cursory overview right now. But if you want to learn more, come to those seminars. And then go to as many seminars as you can. You may look at them right now as just one more lecture, one more hour out of your day that's a class. They're not. They're really not. They're an opportunity for you to learn an incredible amount about some really different fields. And so they're extraordinarily well spent time. So if you get a chance, come hear my talk, come hear all the talks that are going to be going on during the semester. Okay, so one of the projects that we work on in my lab has to do with a group of sponges called clionids, cliona sponges. For those of you writing this down, I will be giving a quiz afterwards, so the rest of you, you're in trouble. The cliona, so this is Clyonovarians, this is Clyonovarians, this is Clyonovarians. Notice that they kind of look different. They all belong supposedly to the same species. That's another question for another day. And this one right here is Clonopulpin tumulus. This is a new species that we actually are in the process of writing up a paper describing. Brand new species that never been described before. It's very cute. I'm going to end with this sponge again. You'll find that I have a real fetish with sponges. I mean, for most of you, I probably have never seen a sponge in the wild, or maybe have seen a sponge in the wild but didn't know what it was. I just really think I don't understand why you're not working on sponges. I just don't. But I'm going to end with this guy, Clonopulpin tumulus. And then there's this little guy, Cervicornia cuspidifera. What color are these?
Yellowish.
Yellowish, brownish, orangish. Granted, my picture taking skills have much to be desired, but they're all kind of brown, yellow, orangish. The reason for that is that they have zooxanthellae symbiont in their tissue. Zooxanthellae are algae. They're protists, little single-celled algae. And they live intracellularly. So they actually occupy a space inside the sponge cell, and they're wrapped by membranes that are derived from the host. So they act a lot like an organelle. Okay? Now, these sponges live in really shallow water, so that lagoon can be as shallow as 20 centimeters. And the forereef, so this is the reef crest, and the waves come crashing in, and these pretty, Finding Nemo. Nemo was right there, swam off, Eastern Australia. How many have seen Finding Nemo? Who has not seen Finding Nemo? Okay, let's mock Jonathan. Dr. Dattelbaum does not. You haven't seen Finding Nemo?
It's such a good movie.
It's such a good movie. It is really a good movie. This forereef right here goes down, 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 down deep, 200, 300 meters. And they have, all those beautiful shots that you saw in Finding Nemo. If you're an algal photosymbiont, so a plant that photosynthesizes, you're going to have lots of light right there, right? If you're at 100 meters on the reef, those light levels drop way, 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 way low. The other thing is that the temperatures in the lagoon go very, very high. Temperatures deep in the reef stay pretty constant throughout the year. So any algal cell that's up here in the shallow waters is going to experience very variable environmental conditions, whereas algae that are down here on the reef, not so much. Yet this guy right here, Cyanivarians formicrustans, is related to this guy right here, Cyanivarians formivarians. And the tricky thing is that they both have the exact same type of zooxanthellae that live inside their tissues. Zooxanthellae are those little plants. So they have the same type of algae. Yet the algae that's here is getting bombarded by light and having temperatures that go, we measured temperatures in one of the habitats that we work in the Keys, that was over 104 degrees Fahrenheit. Now that's, last time you were in a hot tub, if you're ever in a hot tub, that's about how warm your hot tub was. So these sponges are hanging out in the hot tub. Temperatures down here on the reef very rarely get above 87 degrees Fahrenheit, 32, 31 degrees Celsius. That's a big environmental difference for the algae that live in the sponge. So, what does that mean? What we're trying to do is to take different types of algae out of sponges and put them into a new sponge, and then shock those sponges with higher temperatures, higher light levels, lower light levels, and then follow what happens to the symbiosis. So what we can do with these sponges, so we take this sponge right here, this is a normal healthy sponge, and we can carve away that brown zone. That brown zone is where all the algae are found. And you're left with the inside deliciousness. Doesn't that look delicious? I mean, doesn't it look tasty? I think it looks delicious. But you take that little piece in there, and you can get a chunk of what we call aposymbiotic sponge. It doesn't have zooxanthellae. It doesn't have a symbiote. And then you can take zooxanthellae from other organisms, crush them up, extract them, so you get individual cells, and you can put them on top of this aposymbiotic sponge, and after about a week, they start to look like that. They start to reinfect. And my wife, who's on the biology department on our faculty, is an evolutionary developmental geneticist, and she's studying this project. Mallory worked with her this summer. So I was almost ready to pull her down as well. But what April's doing is looking at what genes get turned on when this reinfection process happens. How do these two partners, the zooxanthellae that flood into the cells and then the sponge cells themselves, how do they crosstalk at the molecular level? That's not really what I do. I help, but I do find the sponges. I can get in the water. I'd much rather swim with dangerous animals than be in the lab. I do the lab work regimen. And I'm also not quite so good in the lab. I can do it, not quite as well as I can dive. But what April's doing is trying to figure out what genes get turned on here. How is this symbiosis regulated? I'm more interested in what happens when you mix up different partners. So you take zooxanthellae from this species and you put it in another species. How does that mess things up? And the reason this is important, and you always want to keep track of what the big picture is. How many of you have ever heard of coral bleaching? What is coral bleaching?
Basically, like when the toxins and stuff are in the water, they somewhat, they like get on the corals and it suffocates them, so the coral can take in nutrients or get any source of life from
So why do they call it bleaching?
It's like a blanket, someone taking all of the nutrients and stuff like that.
You're close, Valerie.
The symbionts provide the coloring to the corals, or to sponges. So when the toxins affect the symbionts, so when and then we cause the symbionts to leave. So now you just have the white coral, which is why it's bleached, because bleach can leave this color. That's why.
Exactly. So these coral reefs that are so gorgeous and everything, the brown color of the corals is caused by the algae. Underneath the coral's tissue is a skeleton. It's calcium carbonate, just like chalk, and it's white. It's white as the whiteboard. Now when the animal gets sick because of toxins or pollutants in the water, or waters get too warm, all of a sudden all those zooxanthellae start leaving the coral host, and the color goes from a nice brown to an unhealthy white. So they call it bleaching. Now the problem is that some of those corals die when they bleach. Some of them pick up new zooxanthellae, but that process of picking up new zooxanthellae creates a scenario, an evolutionary scenario, where you actually could favor parasites, cheaters, that don't really like working with that new partner all that well. And so this conflict idea, again, one of the ones that I'm interested in, is when you have partners that are interacting with each other, are the ways that they survive long-term, evolutionarily for the long term, is that they work out solutions to their conflicts, molecular conflicts. It's not like they sit around a negotiating table and say, the zooxanthellae demand the Golgi apparatus. You know, and host cell says, I'll give you part of the endocapillage reticulum, but not the Golgi. That's not the negotiation that we're talking about. Okay? But they do have a negotiation that goes on. How do they reach some sort of détente? Well, so what I do is I stick different types of zooxanthellae into different hosts. And so, working with Dr. Jeremy Weiss, who's a visiting instructor in the biology department right now, we measured, this is a cell. I have a y-axis that is unlabeled. The Dr. Wu's in the back are going like this. They're disapproving of this. This is a stable isotope value. Don't worry about the details of it. But basically, this is the sponge, and this is the zooxanthellae isolated from the sponge. This is the sponge, this is the zooxanthellae isolated from the sponge. This is the sponge, this is the zooxanthellae isolated from the sponge. These are cyanidarians. They have similar signatures for the different types of zooxanthellae. And what that implies is that the zooxanthellae from this sponge actually helps the carbon, what it does is it translocates carbon, so the zooxanthellae photosynthesize and they produce sugars, and then those sugars make their way into the sponge, so they're feeding the sponge. So the sponge's carbon isotope is just like its zooxanthellae. It's like you are what you eat. If you're a vegetarian, your isotopic values are different from a carnivore. Well, in this case, the sponge's isotopic value is just like the zooxanthellae. So that implies that the zooxanthellae are giving up some of that carbon to the host. That's not the case for this species. The zooxanthellae have a signature right here. The sponge is different. This is a new symbiosis. We think this is something that just recently evolved. It's got signs that it's a new symbiosis, evolutionarily speaking. And these guys haven't worked out that détente yet. They haven't figured out how to deal with their conflicts. The sponge is still feeding like a heterotroph, and it's not relying on the zooxanthellae for carbon. If we take the zooxanthellae from this critter right here, and we put it in the sponge right here, all of a sudden that sponge doesn't get carbon anymore. So one of the things we're trying to figure out is when you mix up these partnerships, how does that influence sponge behavior and algal behavior? The other thing that we're doing is we're trying to predict what's going to happen when the waters around coral reefs warm up, as they're predicted to do. So most global warming scenarios, climate change scenarios that look at sea surface temperatures, predict sea surface temperatures are going to go up. So Dr. Dattelbaum already presented some of the data that we're working on in Chesapeake Bay. This is stuff that we're doing down in the Florida Keys. When you rear sponges, and these are all averages without error bars, and I did that for a reason because it gets too messy when the error bars are on there. But when you measure, again, a y-axis without a label, I'm sorry. These are photosynthetic yields. They're a measure of the health of the zooxanthellae. For cyanidarians, formivarians, and cyanidarians, formicrustans, they aren't impacted, at least their algae, aren't impacted by warmer waters. But this unidentified, this Cyanopolden tumulus sponge, when you heat it up, the zooxanthellae crash. Their photosynthetic efficiency just goes to crap. That implies that those zooxanthellae aren't particularly going to do so well when water temperatures go up. The last thing I want to talk about, so that's sort of our symbiosis stuff. The last thing I want to talk about is some work that we're doing with sponge feeding. So if those Zooxanthellae are contributing carbon to their hosts, then, oh no. Oh, I think it's just taking a while to load. If they're contributing carbon to the sponge, then maybe the sponge doesn't have to eat as much heterotrophically. So one of the things that we're studying down in the Keys is we're looking at levels of heterotrophy for the sponges. So we're measuring how much they feed on bacteria that are in a water column. And unfortunately, this isn't going to play. It might start playing in a minute. But what it is a video that shows how we measure pumping rates. These little holes right here are oscula that water comes out of. And that yellow dot up there is a fluorescent dye that actually you can visualize pumping through the sponge body. So that's another project for another day. The last thing that we're working on is a project to describe all of the diversity of sponge life on the planet. And so working with Dr. April Hill again, we're sequencing many genes from many sponges around the world. And it's a project that I'm going to let Michael tell you about. Now, Michael Stefano just joined the lab a month ago, approximately. Whenever the semester began, a week after that, he was in working on the lab. So he's about as new as you can get in terms of doing research, and would be a perfect person to talk to or ask questions about how you get into a research lab. And what's it like? Is it really always this scary? Like you show up in a room, and all of a sudden you're speaking to 30 people about what your research is. 40 people about your research. So Michael, tell them about your project.
What do you want me to tell them?
Tell them about your project.
All right, so I'm working on a project called the Portal Project, which is the Peripheral Tree of Life project. And basically, how old is it? 600 million years old? About 600 million, all of life on Earth we trace back to its common ancestor. And the only ancestor we know before that is sponges, which are 600 million years old. So there's, what are the 4 groups of sponges? I don't know this.
Demosponges, Calcarea, Hexactinellas, and Homoscleromorphs.
So all those 4 that you just mentioned fit into the group Peripheral, and we're going through and trying to figure out how those 4 groups fit together and how all the sponges and species in those groups fit together, and that's all in the Tree of Life. So we get samples from around the world. Malcolm and April they isolate cDNA from the sponges, and my job is to take that DNA and I run a PCR test on it. Or not a test, it's a reaction that basically, let me rewind a little bit. We're looking for 7 genes in those sponges, and those 7 genes are how we're going to look and see how they all fit together. So I take one at a time, one species of sponge, one gene, and I do the PCR reaction, which is something that it's just a little box, and it goes through cycles of hot, cold, hot, cold, hot, cold, hot, cold, over and over and over again. And it basically mimics replication of a certain part of the DNA that you're looking for, which in our case would be one of those 7 genes would be like ALD, ATPB, PFK, there are, I don't know, all 7, but there's a bunch of them. EF1s, version like that. So we take those 7, you basically get the hexagonal together, put it in this reaction, and it replicates that over and over and over and over again. And just so we can try to see if we're looking for that one gene. So to see if we have that one gene, we do a gel electrophoresis. Run it on the gel. You look for that. Basically, you have like a ladder that shows you all these areas. You have a plasmid. It shows you where that gene is on that gel. You look for that gene, and that's how you isolate that gene. Make sure you have the right one, because if you have the wrong one, it messes things up, and you don't want it. It's really confusing. You never know if you mess things up and ruins everything, as I've been told multiple times. And so you have that gel. You can see the bands. We take it upstairs, second floor. There's a machine that runs a UV light through it. We take a picture of it. So we visualize that band. We then take that band, we cut it out, because that's what we want. We want that DNA. And we do the PCR to see that band and to have that DNA. So once we have that band, we clone it, which I haven't done yet. I have not done the cloning. If you're in genetics, we talked about it. You'll see people from genetics class that you're looking at E. coli cells and you have lack of system, and I don't really understand that cloning part.
That's good. You're good. You don't have to go into that kind of detail,
Okay.
but you're good.
Basically, you clone it, and as far as I know, you help me out because I don't know.
Sure.
I just know that you got the blue and the whites, and you pick the whites, not the blues.
So then we take these clones and they have the gene that we're interested in. We isolate that gene through a series of techniques, send it off to sequence. We actually have we're working with the Smithsonian Institution in DC, and they have a very nice sequencing facility. Ship the sequences off. They ship down the DNA sequence to ATGC sequences. Did I forget one? ATGC. Are there any others? No? P's in there, right? No, it's just ATGC. Just checking. It was a little test. I told you I was going to test you. We come back with these sequences, and then we try to fit them together so that we see how those sponges are related to one another on the phylogeny. And the reason that's important is that we don't really understand that the earliest steps in animal evolution. We know that there are these organisms that are very primitive, like the sponge. We don't know how they fit in the animal tree. And so Michael's going to figure that out. Now he's going to kill me tomorrow in lab, but
What else do you want? What else do you want me to say? I don't really know. I'm just trying to walk through the procedure.
that was good. That was
Yeah,
good.
so basically you do the cloning and then
Yeah.